Hello everyone, welcome to Inspire, a short lecture series in science. In short videos, we discuss various topics in science. Today's discussion will be part 2 of our genetics lecture.
In the previous agenda, we saw an introduction to genetics, the what, why and how. We saw a brief history of genetics. Today we are going to talk about experiments in genetics and in particular Mendel's experiments with the pea plant. Before we go there, let's review some terms that we learned previously. We learned about trait and phenotype, which are characteristics under study. We learned about genotype, which is the genetic cause of a phenotype. We learned about lineage, which is a set of mutations or variations that connect an ancestor with a successor. We learned about genetic variations, which describes the alteration in genes amongst members of a species. Variation can be continuous or discrete. Now that we have reviewed what was discussed in the previous lecture, let's go into discussing experiments.
Before we discuss Mendel's experiments, have you thought of how experiments are designed and performed? First, scientists make observations, then they come up with a hypothesis or a model that will fit the observations. This hypothesis is then tested experimentally. For this, an appropriate system must be chosen. Based on this system, they decide the test and the number of repeats for the experiment. This is collectively called experimental design. Good experimental design must always include an appropriate control. The data must be collected and analyzed using statistical methods. Statistical tests must reflect the hypothesis rather than be fit around the data. Mendel's experiments are an example of outstanding experimental design and technique. He chose pea plants with 7 distinct characteristics as shown in the image. So he chose either plant height, which was tall or dwarf, seed color, which was yellow or green, seed shape, which was round or wrinkled, flower color, purple or white, pod shape, which was either inflated or constricted, pod color, which was green or yellow, and flower position, which was axial or terminal. Pea plants were an ideal model system. Here's why. They were easily available, they had clearly distinguishable traits, and they could be easily crossed either by self-pollination or cross-pollination, and they had a relatively short life cycle of 60 to 70 days. Further, Mendel developed pure lines which he would use as control for his experiments. A pure line is a population that breeds true for a particular trait or phenotype being studied. That is, all the offspring produced by self or cross-pollinating within the population are identical for this trait. It took him nearly 2 years to develop these pure lines, and he got 7 pairs of pure lines with each pair differing in only one trait. So we have to appreciate the amount of work he put in to develop these controls prior to starting his experiments. Mendel's investigations into these inheritance patterns used for the first time statistical analysis and mathematical logic to solve problems in biology. His experiments had a large sampling size which gave greater credibility to the data that he collected. Also, the confirmation of his inferences were done by experiments on successive generations of his test plants, thereby proving his hypothesis rather than just leaving them as unsubstantiated ideas. Prior to Mendel's experiments, most people believed that traits in offsprings resulted from a blending of traits from each of the parent. However, in Mendel's experiments, he chose pea plants which had 2 types of flowers, either white or purple. He noticed that the offspring plants did not get flowers with a blending of the 2 traits, that is, lighter purple colored flowers. Instead, they were either purple or white. So Mendel hypothesized that traits must be contributed to by pairs of inheritable elements. He called these pairs of inheritable elements as alleles. Alleles is a word derived from Greek meaning allelomorph, or it means other form. Alleles may be the same, which is homozygous, or different, that is heterozygous. In modern terms, allele is an altered form of a gene. The place in the chromosome where the alleles are found is known as the locus, or in plural, loci. Mendel hypothesized that alleles contributed to traits. Let us take a closer look at one of his experiments with a single trait inheritance pattern. He conducted these using plants that bred true for flower color. In this experiment, Mendel pollinated a uh, purple flowered plant with pollen from white flowered plant. This is the parental generation, which is also represented as F0. The subsequent generation that was generated from this cross is the progeny, also known as the first filial generation or F1 generation. All the plants resulting from this cross had purple colored flowers. The generation of progeny that are generated subsequently by self-pollinating the F1 generation are symbolized by F2, F3 and so on. Mendel also did a reciprocal cross, that is, he pollinated white flowered plants with pollen got from purple colored plants. Interestingly, all the progeny or the F1 generation were once again all purple flowered plants. He concluded that regardless of the type of cross, all the F1 progeny were similar to one type of the parental generation. Next, he observed the F2 generation by self-pollinating the F1 plants. He noted that some of the plants had white flowers. So somehow the white trait had reappeared. He inferred that some whiteness must have been present in the F1 generation but unexpressed. He coined the term dominant and recessive to describe the purple flower plants and the white flowered plants. In other words, the purple phenotype was dominant whereas white was recessive. He noticed similar patterns of dominance and recessiveness when he repeated the same experiments with respect to the other traits. He didn't stop there. Mendel next quantified the number of F2 plants. For example, he counted 705 purple flowered plants and 224 white flowered plants. He noted that the ratio was nearly 3:1 in the F2 generation. So he got the same 3:1 phenotypic ratio in the F2 generations for all the other 6 traits as well. Let us go over the experiment once again. We start with the parental generation or P or F0, pure lines which bred true for a particular trait. In this case, flower color, purple or white. Crossing these pure lines produces a progeny which is referred to as the first filial generation or F1 generation. The F1 progeny were all purple flowered plants. Hence he named the trait that appears in the F1 generation as dominant and the other as recessive. He also did a reciprocal cross which had the same results, that is all purple flowered plants. So he named them dominant and recessive. And the second filial generation, he noted that the white flowered had reappeared and this was in the phenotypic ratio 3:1. So let us pause here and review what we have learned so far.
So we learned about good experimentation practices, the importance of hypothesis testing and the use of controls and a good model system. We saw how Mendel's experiment followed these good experimental practices. And we also saw the Mendelian traits, 7 traits in pea plants. Next, we went a little bit deeper into the trait inheritance patterns using a single trait, that of flower color. We saw that one of the 2 traits disappears in the first filial generation, and that is called the recessive trait. The trait that is seen in the F1 generation is the dominant trait. However, upon further crossing of the F1 generation, we saw that the recessive trait again reappears. The relevance of Mendel's results is that for any discrete trait, these rules that of 3:1 phenotype in the F2 generation hold true. So with that, we will close today and then look at further results and implications of Mendel's work in the next video.
Thank you for listening. If you have any questions, please feel free to comment or email. Thank you.
Have a great day, folks. Bye.